Can everyone hear me? It's good. Thank you for the kind introduction, and thank you to the program committee for inviting me. My lab develops and applies machine learning algorithms to generate discoveries from large-scale brain imaging data. Today I'll talk about the lab's work on using generative models to explore resting state, parcellations, and dynamics. So as we all know by now, the human brain is highly active, even at rest. And furthermore, spontaneous fluctuation in resting state of fMRI can be used to explore brain organization. For example, this is a seed region in superior parietal cortex, and if you take the time course of this seed region and you correlate it across the cortex, you get this network of brain regions shown here. This technique is called function connectivity, and this network is known as the dorsal attention network. Now, if we let the seed region goes around, we can see that the correlation pattern changes a lot. This suggests that adjacent brain regions can have very different connectivity patterns. This has motivated many studies to estimate networks and parcellation from resting state of fMRI, and we can then use these networks and parcellations, or gradients, to explore, to understand brain organization. There's, of course, also a lot of interest in using resting state of fMRI to study neurodegenerative and psychiatric disorders. And furthermore, we can use resting state of fMRI to predict brain maturity and also cognitive abilities in healthy individuals. So overall, resting state fMRI is a powerful tool for exploring healthy brain organization, mental disorders, as well as individual differences in cognition. And so for the rest of this talk, I plan to talk about how we use generative models to explore resting state of fMRI. So the question then is, what is a generative model? So a generative model is a model that can be used to generate observed data. This is obviously kind of a circular definition, and perhaps the best way to explain what's a generative model is that everyone knows what's the most famous generative model, which is Newton's 3 laws of motion. So if we know the initial states of the balls, as well as the force applied by the pool table Q, right, then the Newton's 3 laws allow us to predict how the balls move, although not perhaps the moving of the hole in the table. So this is an example of a physical generative model, but in neuroscience, there's actually a broad range of generative models, right? We can simplify things and say that at one end are biophysical generative models. For example, this might include things like a spiking neuronal network and so on. These biophysical generative models can be highly detailed. For example, a single cortical column can be modeled with thousands of parameters. At the other extreme are statistical generative models. So for example, probabilistic ICA is a statistical generative model. Now, the reason why I like to use generative models is that generative models in general make explicit assumptions about processes underlying the data, and therefore, I believe that these model parameters are more interpretable and can be used to derive insights in the brain. There's a very good article on this by Stefan Hoff in Neuroimage in 2014. I believe it won the best paper that year. If you have not read it, I highly encourage you to read it. So for example, just to give you examples of generative models, so for example, there's been a lot of work that tries to conceptualize hierarchical relationships among behavior, neurocomputation, and neuroanatomy, or task, cognitive processes, and brain activation. Similarly, a hierarchy can be sketched out for brain disorders. This hierarchy shown here is kind of a motivation for the RDoC initiative. So in this illustration, the idea is that a brain circuit supports multiple cognitive processes. Disruption to these processes leads to symptoms. Some symptoms might correspond to diagnostic criteria for disorder A. Some might correspond to diagnostic criteria for disorder B. So importantly, the symptoms can overlap across disorders. So the thing is that these are conceptual models. These are actually not a mathematical model. so we cannot actually fit them to data. So a few years ago, we developed a model, a generative model, that encodes the intuitive idea that different tasks might share multiple cognitive processes, which in turn activate overlapping brain regions. Because this is a generative model, we can fit it to data, and in our case, we fit it to 10,000 experiments across 83 tasks from the BrainMap database. This allows us to estimate quantities such as the probability that a task will create a process, and the probability that the process activates a voxel. Once we have these probabilities, this can provide certain insights to the brain. For example, my collaborator, Maxwell Baudolaro, he shows that complex tasks, so complex tasks are tasks that recruit more cognitive processes. These complex tasks tend to increase activation in connector hubs as opposed to local hubs. So in the case of local hubs, regardless of task complexity, the activation remains relatively constant. So therefore, this suggests that connector hubs might integrate information across specialized brain networks, supporting our ability to execute diverse tasks. Similarly, there's been a lot of work showing that even in typical late-onset Alzheimer's disease, there can be a lot of inter-subject variability. So my student Xu Ming shows that he developed a model that encodes the idea that patients might share multiple latent factors, and these latent factors are associated with distinct patterns of brain atrophy. The key point here is that a patient can express multiple factors to reflect the possibility of coexisting pathologies. So Xu Ming applied this model, fitted this model to voxel-based morphometry data. He showed that there are at least 3 distinct patterns of brain atrophy, and that these patterns of brain atrophy are associated with distinct memory and executive function decline. We're currently extending this model to deal with multiple mental disorders, but I won't have much time to show it to you. If you're interested, you can visit our poster on Wednesday, NUMPO's poster on Wednesday, and Valeria is giving a symposium talk on Wednesday as well. So for the rest of this talk, I'll focus on resting state fMRI. So the rest of this talk is divided into 3 parts. The first part is about individual-specific cortical parcellation. The second part, we will use a simple generative model for fMRI dynamics. And finally, we will talk about biophysical generative models. So in the first part of the work is done by my student and now postdoc, Ruby. So this is the 17 networks from my 2011 papers. These networks are estimated at a group level by averaging data across 1,000 subjects. The problem with group networks is that they do not capture individual-specific network topography. So there's a motivation to estimate individual-specific networks. Furthermore, we will show that this individual-specific network topography can be used to predict behavior. So what's the motivation of this new approach? There's, of course, been a lot of work done on estimating individual-specific networks or parcellations. Across these work, because we're interested in individual-specific networks, all algorithms have to take into account inter-subject variability. But in the case of intra-subject variability, this is usually not taken into account. And the reason why this is important is that the spatial pattern of inter- and intra-subject variability can be quite different across brain regions. For example, studies have shown that the motor cortex exhibit high intra-subject variability, but low inter-subject variability. So what this means is that if you have 2 subjects that have very different motor connectivity patterns, right, this difference might be due to intra-subject variability rather than inter-subject variability. So if the model does not explicitly take this into account, then the model might become confused. So Ruby develops a multi-session hierarchical Bayesian model. This model needs to be trained on multi-session data, but the nice thing is that once it is trained, it can work on with 1 session of data. So let me just give a quick overview of the model. Let's denote subject by S and session by T. I promise there won't be much equations. So the input to this data is are multi-session data from many subjects, right? So for each cortical vertex, we can compute a function connectivity profile. So for example, shown here for PCC, right? And our goal here is that for each, given the observed profiles, we want to assign a label to each vertex, shown here. So then the idea is that vertices with the similar patterns of, vertices with similar connectivity profiles should ideally belong to the same network. Because individual subject profiles can be very noisy, so we have a spatial prior that encourages neighboring vertices to be assigned to the same label. This is actually estimated from data. We have other priors as well. At the top level, we have a group level connectivity profiles, for example, for each network. So for example, here I'm showing the default network connectivity profile. So you can think of this group level profile as the average connectivity profile of all vertices of all sessions of all subjects that belong to the default network. Now to model inter-subject variability, we're gonna have mu-s. So mu-s is the subject-specific connectivity profile. So for example, here's subject 1 default network connectivity profile, and here's subject 2. And you can see that there's inter-subject differences. And this variability is controlled by sigma 1. So larger sigma 1 means there's more variability. Now to handle intra-subject variability, we have mu-st. So mu-st is the network profile of a subject during a particular session. So here I'm showing the default network connectivity profile of subject 2 during session 1. And this is the same subject during session 2. You can see that even for the same subject across sessions, the connectivity profiles can be different. And this variability is controlled by sigma 2. A larger sigma 2 means that there is high variability, high intra-subject variability. And then the sigma 3. Sigma 3 controls the interregional variability. So in our model, we don't assume that a network has a piecewise connectivity, that is a constant connectivity everywhere. So sigma 3 allows us to control how variable is the connectivity within the network. So if sigma 3 is high, this means that within the network, there can be high interregional variability. So we're going to fit all the variables in red from multi-session data, and the nice thing is that once we have fitted the data, we can apply it to a new subject with a single session from a new data set, which is exactly what Ruby did here. She learned a model on the genomic superstruct project data, and she applies it to the HNU data. So the HNU data has 30 subjects, 10 sessions. This allows us to change, to look at how much the parcellations improve as we increase the number of sessions, as we increase it from 1, 2, 3, 4, 5. There's many different parcellation metrics you can use. We actually did a lot of experiments in the paper. I won't bother you with all of them. So for example, we look at homogeneity. So what a homogeneity measure does is that it looks at, it is defined as a correlation between all time courses within the network. So the idea is that a high homogeneity means that the network is more coherent, which implies a better parcellation. This homogeneity is computed on leave-out sessions. Leave-out sessions are sessions that we did not use to parcellate the subject. So the gray bars correspond to Wang and Gordon, and the white bar corresponds to Ruby's approach, and we can see that we have higher homogeneity, suggesting a better parcellation. So as we increase, we can also observe that as we increase the number of sessions, the homogeneity increases for all methods. And so, but you can also see that it tapers out around 4 or 5 sessions. So in this case, it's about 30 or 40 minutes of data. What is cool is that with one session of data, Ruby's approach can generate comparable parcellations with other approaches that uses, for example, 5 sessions of data. The parcellation topography are also highly replicable across sessions. Here are 4 Human Connectome project subjects. Each column is a subject. The top row is day 1, the bottom row is day 2. You can see that there's these network topography that's highly replicable across sessions. Now the question is that, are these real? I mean, these look very nice, but are these real? And so what Ruby did was to show that we can use this topography to predict behavior. Many studies have shown that network size and topography vary across individuals. However, there are few studies showing whether these differences can be used to predict behavior. So we consider 58 behavioral measures from the Human Connectome project. And we're going to use kernel regression for prediction. So what kernel regression does is that we're going to predict the behavior of a test subject as the weighted average of the training subject's behavior. So then the idea is that if the training subject's parcellation is more similar to the test subject parcellation, then we're going to weight the training subject more. The nice thing about this kernel regression framework is then that we can test different hypotheses about what is useful for prediction. So for example, we can define similarity based on topography. So two parcellations are more similar if they have more spatial overlap, or we can do it based on size. Two parcellations are more similar if their networks have similar sizes. And so here is the prediction accuracy for the 58 behavioral measures. Some behaviors are easier to predict than others, so if you average them, we get about 0.13. Just as a sanity check, here's the ACP Megatrol, which uses F's function connected strength for prediction. The results are not completely comparable because there are many variation in how this was done. So I'm not trying to say one is better than the other. What I'm trying to say is that using F's function, using parcellation topography is no worse than using functional connectivity strength. Furthermore, our prediction accuracy is better than using the topography of two other approaches. We can also use network size to predict behavior, but the accuracy is much lower than network topography. So that was the individual specific networks. But we know from system neuroscience that, well, there's no but, there's use for networks as well as this. So we know from system neuroscience that the cortex consists of roughly 300 to 400 cortical areas with distinct function, architectonic connectivity, and topography. So I'll show you Ruby's new work on doing individual specific area level parcellation. So we're going to use the same model essentially with some tweaks. One difference is that we will initialize the training using the Schaefer group level parcellation. This is work done by Alexander Schaefer. So we have previously shown that this parcellation is highly homogeneous in both resting and task data across multiple data sets and pre-processing strategies. So we have tried ICFX, COMCore, global signal regression, and this parcellation is highly homogeneous. This parcellation also aligns well with architectonic boundaries, shown here for area 3, 4, 2, and 17. One thing to note here is that this is not, you can see that the Schaefer parcellation actually subdivide traditional cortical areas. So for example, the face and the hand regions in area 4, right, are separated. So this is why I put quotation mark around aerial, because this is not traditional cortical areas. But I do think that there can be some advantages for this parcellation, because for example, if you're modeling a behavioral task with button presses, it makes sense, for example, to separate the face region from the mouth region. We also have some additional gradient-based spatial priors, which we estimate from data. And the idea of this prior is to ensure that aerial boundary shows sharp transitions in functional connectivity patterns. So again, we trained this model on the Human Connectivity Project and apply it to the Midnight Scanning Club. Here are two subjects from the Midnight Scanning Club. The top row is sessions 1 to 5, the bottom row is sessions 6 to 10. Again, you can see that this aerial topography are variable across subjects, but also highly replicable across sessions. And again, we're going to use them for prediction. So we're going to use, again, 58 behavioral measures from HCP. This is using the network topography I've shown you previously. The numbers are slightly different because now we're using S1200 rather than S900. If we use the Schaefer group level parcellation for prediction, the accuracy is actually better. So the way we do the parcellation, the prediction is that we compute 400 by 400 functional connectivity matrices, and then use that for prediction using kernel regression. If we use an individual specific area level parcellation, we get some improvement. And our approach also shows better behavioral prediction than, for example, a recent paper on individual specific parcellation. So to summarize, I've shown you a multi-session hierarchical Bayesian model, which accounts for both inter-subject and intra-subject variability. The approach actually needs less data than other approaches for equivalent performance. And if we use the same amount of data, the parcellations are more homogeneous in out-of-sample resting and task data. I did not show you the task result. We also show that using individual specific parcellations can improve behavior prediction. And even though the model requires multiple sessions of data for training, but the nice thing is that after training, we can apply it to a new subject with a single session of data from another data set. So let's just move on to the next topic. Next, we'll talk about resting state dynamics and a simple model for resting state dynamics. This is work done by my ex-postdoc, Raphael. So in the previous topic, when we're talking about parcellations, functional connectivity was computed by using the entire time courses. The problem is that this assumes that the functional connectivity is not changing over time. As a result, there has been a lot of work trying to compute functional connectivity over a short time window, such as the one shown here, right? So here, functional connectivity is computed over that small section of time points. We can slide the window and compute a different functional connectivity matrix, right? And we can keep sliding the window until we have many functional connectivity matrix. Now, this is called time-varying functional connectivity, sliding window functional connectivity, and so on. There are many, many ways to do this. I'm just showing you one of them. Once we have all these sliding windows correlation matrices, what are we going to use them for? I feel that one nice way of visualizing all these matrices, because there are many matrices, is to compute these functional connectivity dynamic matrices, right? So what this does is that we compute correlations between all pairs of these sliding window connectivity matrices. So shown here. So the matrix below, that is a T by T matrix, where T is the number of sliding window. You can see that there's an interesting pattern in this functional connectivity dynamics matrix. For example, the diagonal is all red, which is a result of autocorrelation in fMRI data, and because of overlapping sliding windows. What is interesting is the off-diagonal patterns. There seems to be these blocks of recurring connectivity patterns. And this has intrigued quite a number of people in the community. So for example, the question would be, are these blocks of recurring connectivity patterns indicative of discrete brain states? So one way to look at this problems would be to fit different generative models to the fMRI data, right, and try to see which generative model might explain this functional connectivity dynamics better. So for example, people have used autoregressive model to fit to fMRI data, right? So in an autoregressive model, what this is saying is that the fMRI data at time t+1 is a linear combination of the fMRI data at the previous time point, plus some noise. So there have been studies that fit these autoregressive models to pairs of ROIs, pairs of brain regions jointly. That fits this bivariate autoregressive model to each pair of ROIs independently, right? And if you do that, you get this functional connectivity dynamics matrix shown here. And you can see that the off-diagonal is very flat, and there is no recurring connectivity patterns. On the other hand, if you fit a hidden Markov models, right, you then get these blocks of recurring connectivity patterns. And so this has been interpreted as some evidence for discrete brain states. Although one thing we should note is that you can see that with the hidden Markov model, we tend to get these very sharp transitions not seen in real data. So what Raphael did was that instead of fitting a bivariate autoregressive model, he fitted a multivariate autoregressive model. So the only difference is that in the multivariate autoregressive model, we fit the model to all brain regions jointly, right? So in the bivariate autoregressive model, we fit it to each pair of brain regions, right? While in the multivariate autoregressive model, we fit it to all regions jointly. And we can see that with the multivariate autoregressive model, we can actually generate very good functional connectivity dynamics. What is the implication of this? This is kind of controversial, so I just want to be clear, right? I'm not claiming that there is no discrete brain state, right? I'm also not claiming that hidden Markov models are not useful. In fact, we have been involved in papers that use hidden Markov models. What I'm saying is that discrete brain states are not necessary to generate realistic functional connectivity dynamics. Now, in follow-up work, Raphael shows that the multivariate autoregressive model can explain behavior better than static function connectivity. And what he then do next is that he sorted the 58 behavioral measures. Again, this is from the Human Connectome project. He sorted the behavioral measures so that on the left, the static function connectivity is better than the autoregressive model, and on the right, the autoregressive model is better. He then colored the different behavior. So he colored the self-reported measures in orange and the task performance measures in green. I just realized that you might have problems if you're colorblind. I'm sorry about that, yeah. But trust me that the orange self-reported measure seems to be clustered to the left. So indeed, if we take all the self-reported measures, we see that there's no difference between static function connectivity and the autoregressive model. But if you look at task performance measure, the autoregressive model can explain these measures better. So to summarize, static function connectivity does not account for temporal dynamics. Using sliding windows correlation and all sorts of other approaches, right, we can explore these temporal dynamics a bit better. The interesting thing is that the multivariate autoregressive model does not assume discrete brain states, but it can generate very realistic functional connectivity dynamics. Raphael also shows that multivariate autoregressive models can explain task performance better than static FC, function connectivity, right? And self-reported measures are explained equally well by both. So therefore, resting state brain dynamics at different time scales can capture distinct aspects of human behavior. So although the autoregressive model can capture resting state dynamics very well, it is a statistical generative model. As a result, it can be hard to try to derive mechanistic insights from it. So therefore, a few years back, I was getting very interested in biophysical generative modeling. And today we'll talk about our work on neural mass modeling. So this is work done by my postdoc, Peng Wang. He looks very serious in this photo, but he's actually quite a laid-back person, yeah. So as we all know, the brain is organized at multiple scales, right? From genes, neurons, all the way to large-scale brain systems. However, we can study the brains at different spatial scales, but how these different scales interact is still relatively not as well studied. One way to bridge this micro scale and macro scale gap is to use biophysical modeling. As I was telling you earlier, for generative models, we can kind of put them on a spectrum. On one hand is the more biophysical generative model, and on the other end is the more statistical generative model. So today we will talk about neural mass model, which is a simplification of spiking neuronal models. So it is not super biologically realistic, but it will be more biologically realistic than, for example, the autoregressive model. And there's been a lot of previous work, of course, on using neural mass model to study resting state fMRI, providing mechanistic insights into resting state fMRI. Before I talk more about neural mass model, let me first talk about the framework for studying neural mass model. So usually in this kind of studies, we start off with a structural connectivity matrix. This structural connectivity matrix can be from diffusion MRI, or it can be from invasive track tracing studies. So given the given the structural connectivity matrix, we can then apply a neurodynamic model to this to the structural connectivity matrix. This allows us to generate simulated neural signals, right? We can then apply a hemodynamic model, which allows us to generate simulated function connectivity data, right? So in this kind of model, we don't usually consider task inputs. So as a result, we consider these to be simulated resting state function connectivity. And what people do is that we then compare it to empirical, real resting state function connectivity. And the key assumption is that if the real function connectivity and the simulated function connectivity matches quite well, right? Then this suggests that the model is a good representation of the brain, and then we can try to interpret the model. So most people just use the balloon model, and this is what we'll use here as well. But of course, you can use other hemodynamic model. Today, we'll focus on the neurodynamic model. There are, in fact, many models we can use. Here, we'll focus on the dynamic mean field model by Gustavo Deco. So, one second. So what the mean field model does is that it's a mean field reduction of spiking neuronal networks into a more simplified model. So in the spiking neuronal networks, they are coupled by AMPA, NMDA, and GABA receptors. But this makes things rather complicated, and therefore Gustavo and others have simplified these spiking neuronal models to neural mass model. These neural mass model can capture less biologically realistic, of course, but they're able to capture large-scale brain dynamics with relatively low parametric complexity. So in a neural mass model, what we do is that each cortical ROI is represented by a system of nonlinear ordinary differential equation, coupled together by the structural connectivity matrix that I was showing you before. So the dynamics at each ROI, at each regions of interest, is driven by 4 input types, right? The first input is the recurrent or the intra-regional connectivity, right? This is parameterized by the recurrent connection strength W. The second input is inputs from other brain regions, right? So this is parameterized by the structural connectivity matrix, scaled by this global constant G. The third input is the external input current. The external input currents can be thought of, for example, as potentially inputs from subcortical relays, and it can also be thought of as a term to capture essentially things we're not modeling. And the fourth input is neuronal noise, parameterized by the amplitude sigma. So Gustavo Deco and others have shown that we can achieve good agreement between simulated and empirical function connectivity when the brain operates close to the edge of criticality. I'm always very jealous of these computational neuroscientists with cool terms such as criticality. Yeah. But yeah, it's great. Anyway, so the tricky situation is that in this kind of neural mass model, the parameters are usually manually set. So in this case, the recurrent connection strength, the global interregional scaling constant G, the external input I, and the neuronal noise amplitude are manually set, right? As a result, you have to make simplifying assumptions. For example, we assume that the recurrent connection strength and the external inputs are the same across ROIs. The problem is that this is not very biologically plausible because we know that different brain regions have different cytoarchitecture and so on, right? But this is necessary because the 4 parameters are manually set. So if we allow each brain region to have their own microscale parameters, then the problem quickly becomes intractable. So the goal of our recent work is to try to automatically estimate the mean field model parameter, right? So this allows us to relax the mean field parameters so that the recurrent connection strength W and the external input I are different across cortical regions. So in the end, we optimize 138 parameters using a modified EM algorithm. It's basically similar to what Carl Friston used for DCM. And what we do is we maximize the fit to the static function connectivity. So we use the human connectome project. We divide the data into training and test set. We fit the parameter using the training set, and then evaluate the fit using the test set. Now, in the test set, what we find is that the agreement between the simulated function connectivity and the real function connectivity is about 0.48. If we use the original mean field model, so the original mean field model is the one where we assume that all microscale parameters are the same across brain regions. The agreement between a simulated and real function connectivity is only 0.27. And then if we do a control analysis where we correlate the structural connectivity with empirical function connectivity with, is only 0.3. It's actually 0.3. So if you compare 0.3 and 0.27, this shows that actually the original mean field model, at least in this data set, is not doing much besides recapitulating structural connectivity. This is actually an important control conditions, but many studies actually do not report the correlation between structural connectivity and empirical function connectivity. And the reason why this control analysis is important is because you want to make sure that your dynamic model is doing something beyond just recapitulating structural connectivity. So then the next thing we were interested in is, we allow each brain region to have different macro-scale parameters, right? So I'm wondering, is there a meaningful spatial patterns? So here I'm showing you the recurrent connection strength, W. And we can immediately see that that's actually a very, very beautiful pattern, right? The black boundaries on this figure here corresponds to the boundaries from my 7 network parcellation, right? If we focus on the yellow arrows, right? The yellow arrows are pointing to early sensory motor regions, including auditory cortex, somatomotor cortex, and visual cortex. The arrows are pointing to purple regions, which means that it has high recurrent connection strength. On the other hand, the arrows are now pointing to default network regions. And on the left, you can see that the arrows are pointing to a light blue color, which means that it has low recurrent connection strength. Which means, so the default network has weak recurrent connection strength. So this is the recurrent connection strength. What about the external inputs? So this is showing the map of the external inputs. And again, we can see that there's a nice spatial topography. So sensory motor regions have strong external inputs, as you can see here. As you can see by it's dark purple, right? On the other hand, the default network has weak external inputs, right? So what I've shown you is that even though the mean field model does not assume the existence of a cortical hierarchy, but we can see that the estimated model parameters review a large-scale cortical gradient. And so what we did was, let's compare it with Daniel Margulies' principal gradient. So we take Daniel's principal gradients shown on the left, we can see that it's actually correlated with both recurrent connection strength and external inputs. We also take cellular density estimates from Martin van der Heuvel, right? And we found that the recurrent connection strength is correlated with neuronal density, right? But not so for external inputs. And then we also look at cell size. In this case, cell size is not correlated with anything. Now, this is all good, but we were wondering, as a follow-up to Wang Peng's study, we were wondering if the relaxed mean field model generate realistic function connectivity dynamics. And just to remind you, this is the same subject I've shown you before, which has this very rich function connectivity dynamics. This is the multivariate autoregressive model. It also has rich functional connectivity dynamics. And then this is the mean field model. So you can see that the mean field model results in this very flat functional connectivity dynamics with no recurrent connectivity patterns. This is very sad because the mean field model is more complex, it's supposed to be more biologically realistic, but it does not explain the fMRI dynamics as well. So there's an important lesson here, which is that more complicated models might not be better. So actually the problem here is that we overfitted to static function connectivity. So my student Xiaolu then changed the algorithm, right, so that we fit to both static function connectivity and functional connectivity dynamics. And now we see this rich functional connectivity dynamics. And the cool thing is that even with this new algorithm, we see these large-scale gradients like before. Okay, just to summarize, I've shown you that we can automatically fit neuromass models. This allows for more biologically plausible mean field model with region-specific properties. This allows to dramatically improve the agreement between simulated and real functional connectivity without assuming the existence of a cortical hierarchy. The model parameters actually review a large-scale cortical gradient. Of course, the idea of a cortical hierarchy is not new. There's been a lot of work, for example, in the monkey and primate literature from David Van Essens and others, right? there's also been work done on this by Daniel Margulies as well as John Murray's group using imaging data. What I'm very happy about this work is that we were able to use a computational model to link microscale properties of the brain with large-scale gradients. We found that the sensory motor regions have strong recurrent connection strength and high cellular density. It also has strong external inputs, which might be related to subcortical information. On the other hand, the default network has weak external inputs, which might be consistent with its potential position at the apex of the cortical hierarchy. So to summarize, I have shown you generative models of cortical parcellations in dynamics. In some sense, we can divide generative models as, we can use generative models as models for analyzing data. We can also use generative models as models of the brain. An important lesson which I want to convey, and which I always forget myself as well, which is that complex, more realistic models are not necessarily better. In fact, a simple autoregressive model, right, can generate realistic fMRI dynamics as good as the more complex, more biologically realistic neuromass model. This is, in fact, a lesson that I always try to remind myself. And in fact, before I end, this lesson applies to a lot of things. As you all know, there's a lot of interest in deep learning now. But we can see, for example, in the recent ABCD IQ challenge, where the goal is to predict IQ from structural MRI data, actually, kernel regression was actually better than deep learning. In our own work, using the UK Biobank, using up to 10,000, 20,000 subjects, we showed that if the goal is to predict behavior using static functional connectivity, right, then kernel regression can perform as well as various deep learning algorithms. And maybe we're just terrible at fitting deep neural networks. But in Google's own study last year, actually, they showed that basically logistic regression is as good as deep learning. This is, if you look at the bottom plot, the top row is deep learning, and the second row is logistic regression. And you can see that logistic regression, the confidence interval are completely overlapping. Again, I'm not saying that deep learning is not useful, right? We're also doing deep learning. Okay, everyone's doing deep learning, right? But, right, and furthermore, it's obviously the case that in some application, deep learning is definitely better, right? But I just want to emphasize that in other applications, right, maybe simple methods can be just as good, right? So feel free to try linear regression, or kernel regression, like it's totally fine, yeah. Anyway, well, I mean, a lot of the AI companies, as you all know, from what I read from the media, is a lot of the AI companies are actually, using linear regression anyway. They just market themselves as using, AI, right? So in general, my lab put all our code and data or parcellations available. I don't collect data. Yeah, I'm a leech, data leech, yeah. So we usually make our code publicly available. So this includes my 2011 parcellation and code, Alzheimer's disease subtypes code, Raphael's multivariate autoregressive model, the different Schaefer group level parcellation, Ruby's individual specific parcellation. We recently did some work on mapping between MNI 152 and FS average space. I know that a lot of people map their data to MNI space and do the analysis there, and then after that they want to visualize on the cortical surface. So we actually provide tools to allow you to map between MNI 152 and FS average surface. But just to be clear, of course, that's what the review complained as well, is that the best way to visualize data on the FS average surface is to project your data to FS average surface and then do your analysis there. But I'm sure there are people that love doing analysis in MNI space and want to visualize that data on FS average afterwards. So we're helping you do that. So anyway, I'd like to thank my funding and support. I also like to thank my lab members, past and current. The one in blues, I've shown have helped contributed towards this presentation. I also like to thank all my collaborators. That's all. Thank you. I think that's the end of the session. We can party now.